Hey everyone! In the last video, you learned how to describe a locus in a genome in a couple of different ways. In this video, you'll see that what we call a chromosome can actually change as we proceed through cell division. This can get a little confusing, but hang in there because it will become important once we start talking about mitosis and meiosis. Now in this video, we have two questions.
And our two questions are, what are DNA molecules and groups of DNA molecules called at different stages of cell division? And which DNA molecules have the same genes and which have the same alleles of those genes? To answer these questions, we're going to follow what happens to two chromosome types within a cell. We'll be following only chromosome 1 and chromosome 20 through cell division, although the same things are happening to all the other chromosome types as well. At this point, each chromosome is made of one DNA molecule. Let's follow the alleles of three hypothetical genes. Two of them, the A and B genes, are on chromosome 1, and the third, gene C, is on chromosome 20. Let's say that this cell is a trihybrid, so it's heterozygous for all three of these genes. So the genotype of this diploid cell is A1A2, B1B2, C1C2. Keep in mind, though, that each chromosome has many genes on it. We're just following three of them here. The pair of chromosomes of the same type, for example, these two chromosome 1s, have the same genes. They are therefore called homologous chromosomes. The prefix homo meaning same. Chromosomes of different types, such as chromosome 1 and chromosome 20, have different genes and are called non-homologous chromosomes. Before a cell divides, it must replicate both chromosomes of each chromosome type. As replication occurs, an exact copy of both chromosomes of each type are made. Each newly made chromosome remains attached to the original at the centromere. This structure, made of two connected DNA molecules, which are exact copies of each other, is considered one chromosome, even though there are two DNA molecules in it and two copies of each gene. So, before the chromosomes are replicated, a chromosome has one DNA molecule and one copy of each gene. After replication, each chromosome has two DNA molecules and two copies of each gene. Because they're exact copies, these two DNA molecules have the same genes and the same alleles of those genes. This structure is called a replicated chromosome. And the same is true of the other chromosomes in the cell. Both of our newly replicated homologous chromosome 1s have the same genes, here the A gene and the B gene, but they don't necessarily have the same alleles of those genes. This chromosome 1 has the A1 and B1 alleles, while this one has the A2 and B2 alleles. Of course, if this organism was homozygous for the A and B genes, then the homologous chromosomes would have the same alleles of these genes. So homologous chromosomes can have different alleles of a gene, but only if the organism is heterozygous for that gene. As the cell starts undergoing cell division, the chromosomes condense. These are still replicated chromosomes. These are still homologous chromosomes, and these are still non-homologous chromosomes. These condensed replicated chromosomes contain 2 DNA molecules, and because they are now condensed, they are referred to as chromatids. The 2 chromatids in the same replicated chromosome are called sister chromatids and have the same genes and the same alleles, because one chromatid was synthesized using the other as a template. This one chromosome is made of 2 sister chromatids. This is true for all of the chromosomes at this point. So we see 4 chromosomes in this cell. 1, 2, 3, 4. The chromatids of homologous chromosomes are called non-sister chromatids. These have the same genes, but not necessarily the same alleles. When we are looking at a karyogram, keep in mind that we are looking at replicated chromosomes, and that each of these chromosomes has 2 DNA molecules and 2 copies of each gene. During meiosis, which we'll examine later, homologous chromosomes can come together to form a structure with 4 chromatids total. This is known as a tetrad, tetra being the prefix for 4. A tetrad is comprised of 2 chromosomes and 4 chromatids. When homologous chromosomes are not in tetrads like these, they are called dyads. Each dyad is 1 chromosome, has 1 centromere, and has 2 DNA molecules and 2 chromatids. Here we have replicated chromosomes 1 and 20. In this picture, we see 4 total chromosomes and 8 DNA molecules. During some types of cell division, sister chromatids of each replicated chromosome are pulled apart. Because they're no longer attached at the centromere, each of these sister chromatids is considered a chromosome. So we now have 8 chromosomes instead of 4. So the number of chromosomes in a cell actually increases when this occurs, even though the number of DNA molecules, in this example 8, stays the same.
Okay, so at this point you should be familiar with the naming of chromosomes and DNA molecules at different points during cell division. If you need a little more practice, feel free to watch the video again. Now, in the next video, we'll take a look at the number of chromosomes, DNA molecules, and gene copies, and how those numbers change as the cell goes through cell division. See you then.